Our first speaker is Mukesh Verma. He's from the DCCPS, Epidemiology and Genomics Research Program. He came to NCI in 2004 from George Washington University. I met him there, and I've known him for about 20 years. And he first came and was in charge of the epidemiology research branch. And he worked also in the Division of Cancer Prevention in the area of biomarkers. And he's also done stuff with the Small Business Innovation Research and Small Business Technology Transfer Programs. And he's going to talk to us today about epigenetics, and cancer control and prevention. Are we ready for the prime time? We'll find out.
Yeah. Thanks a lot, Dr. Modi. So I'm glad to speak about epigenetics, and during my discussion or presentation, I will be giving what kind of research we have done, which is applicable for cancer control or prevention, and where we stand. So about cancer, as you know, that a lot of survivors are there. This is such disease. In some organ sites, we have made some success. In others, we have not. Prostate cancer is the #1 in men, and breast cancer in women. In 2010, number of survivors were about 14 million, but it is expected that by 2022, 18 million cancer survivors will be there. And in terms of cancer control or prevention, we follow all those approaches like early detection or diagnosis and treatment or follow-up of treatment and end of life. And if you consider prevention, it can be either before disease has happened or cancer recurrence. If we can stop that, that is also prevention. Or secondary cancers don't happen, that is prevention. So prevention could be restoring transcription or halting progression or stopping of metastasis. This is general slide for major cancers, which are breast, cervix, colon, prostate. Many years it takes by the time cancer develops. So our idea is that if we can focus on early stages and identify biomarkers or other criteria, then we have more chances for treatment or other options of control. So as it has been shown in colon cancer, genomic mutations, those were identified in those cases, and depending on the age, they were appearing. So genetic part is quite developed, and a lot of years and efforts were made in that. And here it is shown by your age, what name we give for that disease. And those are the diseases where 18-20 years it takes by the time it develops. Coming to epigenetics, these are such alterations where nucleotide sequence don't change, but gene expression you can change. They are needed for normal development, but in any abnormal situation because of exposure, medication, or age, that can lead to abnormal development and abnormal gene expression. You heard about genetic code that you know, but in epigenetics, 2 more codes, methylation code or histone codes are there. In general sense, if you want to know about epigenetics, so genetics is your capability or what you are capable of doing, but epigenetics is the one which regulates that when something should be started and when a process is stopped. That is epigenetics. And a balance of genetics and epigenetics is there during normal development. You don't have to understand epigenetics to know about genetics, but if you want to know epigenetics, then genetics should be known. So if you see the development, how it has come initially, like protogenetics, that came Mendelian inheritance and other processes. Then age of genetics came when several mutations were isolated and disease were associated, basically found out based on that. Then structure of DNA or genome sequencing, that was done. But after 2000, epigenetics has come out. And the reason is When mutations or the SNPs or those changes, alteration in genomes, whether deletion, addition, or copy number changes, they were seen. Many times those disease variations were not located in the coding region. They were somewhere else. And whether those changes are there or not, in some cases diseases were developed. And that's how epigenetics came into picture. So little bit I will tell you about what we know in genetics and how we are taking advantage of that to utilize in epigenetics. In genome, we know for most of the organ sites which are the major mutations. Similarly, we know about 11 or 12 pathways are there. They are the major pathways which have been identified by biochemistry or genetic analysis. And when genomic sequencing came, then for chromosome also, for different locations, the variation in different diseases were identified. When genomic sequencing was going on, then based on HLA typing and by genome-wide association studies, these were done. And then as you can see that on all chromosomes, most of the mutations have been identified where they are located, and that helps us if we see some changes inherited or somatically acquired, that helps in designing our approaches to treat that disease. About epigenetics, after learning more, then we know that DNA is not the only one which will contribute to the disease, but in epigenetics, and initially how it was proved that monozygotic twins, cohorts of that was taken, and they were followed that how much is the concordance and how much is the discordance. So initially it was for cardiovascular disease, but later on in cancer and other diseases also it was observed. And since in monozygotic twins, genome is the same, but still those twins develop different diseases, that gave an idea that environment, lifestyle, or other factors are also there which contribute more. So nowadays we say that there is more to the genome than its sequence itself. So we finished at NIH, as you know, human genome map, and epigenomics we did 2-3 years ago. I will show some data. And now we have started one 4D nucleome where real-time chromosomal structure and DNAs were hypersensitivity sites and other things you can follow. And these kinds of articles also time to time come, like we are in a stage of epigenetic development, epigenetics in the driver's seat. Giving a little background about chromatin or nucleosome. In chromatin, protein and DNA are there, and chromatin, those histone proteins are to neutralize charge of DNA. 30 nanometer is the size of this chromatin, and if you see about 2 turns of DNA, 146 base pair, they will be in nucleosomes. And histones are of 4 kinds, S2A, S2B, S3, and H4. H1 is just a linker histone. And these are octamers. And in 1974, Roger Kornberg discovered these things about nucleosome. So their location also decides whether gene expression will be there or not. After that, one phenomena was observed. In the promoter region, basically, initially it was observed that CG sequences are there tandemly. So if in one region, in 0.4 kb, you have 40 to 60% CG sequences, then generally see that cytosine at 5' position, it gets methylated. So normally C will bind with G, but it starts behaving like U, so it will bind with T. And in next replication cycle, disturbances will be there. So this phenomenon is called hypermethylation, and those sites were called CpG-RLN. At the same time, and this was observed in tumor suppressor genes, and at the same time in some genes, promoter was already methylated, it got hypomethylated, so these cytosine, they lost their methyl group, and gene get inactivated, which was an oncogene. So whether tumor suppressor gene or oncogene, hypermethylation or hypomethylation, they contribute to cancer development. These are the components of epigenetics. Methylation, I told you, in which DNA methyl transferase, they also have different kinds. When methylation starts, then for initiation, you need different methyl transferase than for maintenance of methylation or other processes. Modification of histones are several. Similarly, non-coding RNA, especially microRNA or long RNA, they come under this regulation. Some other terms people have started using like shores and shelves and OpenSea. So shores are those which are less than 2 kb from main CPG Ireland, where Tata and Cat boxes are there. Shelves from 2 to 4 kb, and other region is called OpenSea. So this is the nomenclature I wanted you to introduce. Epigenetics is affected by several factors, whether environmental and genomic or others. And this I told you about hypermethylation and hypomethylation. Histones, two major enzymes are histone acetyltransferase or histone deacetylase. So addition of acetyl group or dissociation, that also contributes to that. And other modifications of histones are shown here. I'll tell you about little bit about microRNA regulation also, but some non-histone proteins like polychrome group of proteins, they are also contributing in regulation epigenetically. And in certain situation of specific histone modification, when in those modification, EZH2 another either one factor or several factors come, which have become now target, which people can target to develop drugs. Sign and line sequences, which are repeat sequences, they are hypermethylated from the start. With age, they get hypomethylated. They are required for the stability of genome. In histones also, three terms you will be seeing: writers, readers, and erasers. Writers are like methyl transferases, readers such as methyl DNA binding proteins, or erasers like deacetylases. So they work in coordination with methylation. So this I told you that by doing several twin studies, it was found that in different diseases, either genetics plays a major role or epigenetics plays a major role. So in different diseases that has been done, and cancer comes in such a way that in this lifestyle and environment, they play a major role than the family history. And all those lifestyle or diet or nutrition and exposure to whether infectious agents or radiation or insecticide, pesticide, they are the major contributors in environment. Other point is that at early stages, after fertilization or very early pre-peri and postnatal stages, that time chances of mistakes are more, and that is the time after genomic sequencing was done and epigenomic sequencing or profiling of all those components were done, that during that time, genetic changes are not there, but epigenetic changes start. And later on, those are less. And in this slide, I have shown the different kind of biospecimen you can collect at different stages, at early stages or later stages, which are suitable for epigenetic analysis. And when I say epigenetic analysis or profiling, it includes all four: methylation analysis, histone modification, non-coding RNA or microRNA profiling, and chromatin compactability or DNase 1 hypersensitivity site. So those can be done. And you can think if some mother is pregnant, then, and if she is smoking, then not only her life is affected, but fetus and stem cells within that. So they are also affected. That way, 3 generations are affected. And we emphasize so much about early life exposure or early detection because of that. And one example is here, like aldehyde and nitric oxide, which are present in cigarette smoke, they induce phosphorylation of histones. So those kinds of studies have been done. And nowadays, big project we are supporting where effect of e-cigarette vapor that we are studying, but results have not come. And these are some of the publications later on you can read, like maternal smoking during pregnancy, that is affected, but methylation is the mechanism. Similarly, maternal anxiety also affect that, and here methylation of specific sites and histones are involved. And to identify those people who are at high risk of developing cancer, then we do epidemiological studies. And we call it cancer etiology, or those factors which are affecting the disease. For that, what we need such population which is disease-free and we can follow for a long time. And we should know about their exposure, family history, diet, medication, etc. and then easily collectible biospecimen, mostly non-invasively, and then follow up. Multiple times we have to take sample so that suppose disease is developed and those samples are also stored, we can go back and see when the first marker or first sign of the disease we see, and that helps generally in developing therapeutic or intervention agents. What is the challenge? These are very expensive, and the data storage or data sharing, in that we are facing some challenges, but advantages that these processes or these steps are essential to identify high-risk factors. Epigenetic biomarkers, which we say that are mostly environmental inducible, and genome we sequence one time because all cells have the same genome, but epigenome has different sequence, so they are cell-specific. Factors can be exogenous, like lifestyle factor I described, or they can be endogenous, whether it is stress, inflammation, metabolic disorders, or hormones. Our program is epidemiology program, so there what we do, we support several cohorts in which people from different countries are there, and those people have not developed disease, but we follow and collect biospecimen and see when they have developed disease. Cancer we follow, but we follow for other diseases also, so that we know what comorbidity or other diseases also developed by that side. And about 2.3 million subjects have been enrolled. We support cohort as well as registries, colon cancer or pancreatic or breast cancer registry. So 62 cohorts we support, about 4 million, and these are the names like nurses health study, physicians study, multi-ethnic cohort. And membership we give if we have more than 10,000 samples at the same time. Why we do that? So that we can study genome-wide association studies, so that we can identify cancer susceptibility regions and understand gene-gene or gene-environment interaction. Many times blood is the only sample which we collect, but that helps us to systematically follow inflammatory response, or at least we can get those changes many times over time in the same sample, and that gives good profiling for us. And time to time we write articles showing that what is the potential of these technologies or approaches and what is happening, and this is about early life exposure of infectious agent and cancer development that we did. And at NIH, one program was initiated about 3 years ago. It is called Environment and Influences on Child Health Outcome, ECHO. This was the program related with children's development, because neurological diseases, asthma, and inflammation and obesity, all those are there. So in that program, all environmental components, those are being measured, and 50,000 pregnant mothers, they have been enrolled in 84 cohorts. So idea is, and this is a major program I am telling, because about $165 million per year we spend, and for 7 years it is congressionally mandated or dedicated amount is there. And to organize that, we have different components like coordination center or cohort sites or genetic core or patient reported outcome. All those are together, but main emphasis is that preconception, those information about mother and father and biological samples are collected, then perinatal, labor through discharge less than 1 month, or infancy after child is born 1 month to 11 months. So very early stages, all those consecutively on 50,000 children, those will be followed. And then genomic analysis, epigenomic analysis, what environmental factors are there, transcriptomics or metabolomics, all those will be done. And then data will be integrated to see what are the factors contributing to those diseases. You hear about children's leukemia or other cancers also, which develop in early stages. So its advantages that these are called longitudinal cohorts, where we follow same person several times. And generally placenta, cord blood, nail, hair, saliva, urine, maternal blood or milk before and after pregnancy, that is done. And such techniques in NMR or mass spectrometry are there, that if you take nail or hair, they can tell what kind of exposure was there. So those exist. Now, epigenetics is affecting behavior also. Previously, they thought that only cancer kind of disease, means different factors only, such factors like lifestyle or nutrition we can do, but that also has been observed, like anxiety during pregnancy that affect methylation, and what kind of biospecimens are suitable. So several of them have been done. Previously, only 3-4 were there for epigenetic analysis. For aggressive behavior also, the way we do genome-wide association studies, where variants are studied with respect to disease development, similarly, epigenome profiling has also been done. And in this report, it was shown that aggressive behavior also has epigenetic regulation. And one article I wrote, which was biopsychosocial pathways. So we try to identify those pathways. And one area is coming which we are promoting very much. It is called social epigenomics, because it starts with where you live, what kind of food or nutritious food are available, how much is the medical facility are there. So they also affect your histone profiling or methylation, etc. So such projects we are funding, where people have access to those population and they can take several samples. At the same time, they are capable of doing either methylation analysis or histone or microRNA or all. And in this article, I wrote that GWAS and EWAS, which we call epigenome-wide association studies, they should go together, because then we can conclude better about sensitivity of people. In the environment, if you take arsenic, benzene, cadmium, chromium, nickel, all those compounds, now publications are coming, they are regulated epigenetically. And since these slides are with you, later on you can read about that, or you can call me, I can explain more. Cross-generational effect of some changes have been seen, like here is alcohol dependency. And that gave rise to another area of research, like toxicogenomics, like all people don't respond to the drug based on their genomic profiling. But now we are studying that epigenomic profiling also affect that. So that area of toxico-epigenomics is coming. On this slide, I want to show that anytime exposure is there, then transiently or in few minutes or half an hour or 1 hour, transcription factor or cell cycle regulatory changes you can see. If that exposure is for a little longer time, 8 hours, 10 hours, or 1 day, then methylation profiling or chromatin changes that can happen. It is only after very long exposure, then only these genomic changes come. So most of the time, if we can avoid that, then a lot of things can be done. Here we have shown that in terms of genomics, how many translocation or deletions or amplifications were done. Similar thing we want to do for epigenetics also. In your mind, you can see that between genotype and phenotype, 2 more things are there, epigenotype, and then metabolism happen, then stable or unstable metabolites come, then only they give rise to phenotype. Phenotype means like disease-specific features. Now those component of epigenetics or epigenetic modifier, they are also coded by some genes. So it was observed later on that some mutations are in those genes, and in this slide I have shown, like these are epigenetic modifiers, as I mentioned, and these are the genes where mutations were found. So we are also trying to find out how much genetic and epigenetic crosstalk or interaction is there. In epigenetics regulation, first histone modification starts, which inactivates or deacetylation of histone. Here it is shown for lysine 9, but if that gets methylated mono, di, or tri, then it gets trimethylated and it becomes stably silent when DNA methyltransferase comes. And Peter Jones and Andrew Feinberg, they were the pioneers in showing these steps. And to understand it more, if you see chromatin, and these nucleosomes are quite apart, acetylation is shown in yellow. If you see at the end, these chromatin are very compact. These are transcriptionally active, but gradually methylation starts, which are shown in red, and yellow deacetylation starts, and finally many factors, such factors come, the transcription cannot happen. So those steps have been identified, and methylation also happens one strand at a time. So once the sequencing is done, then both of the strand should be sequenced to get the accurate results about methylation. Generally, global hypomethylation is there for most of the genes or genome. Then in promoter region, mostly hypermethylation is there, and histone modification keep changing with time or with age. Line sequences which I mentioned, these are the repeat sequences or ALU sequences. So they are hypermethylated already, but with the time they get hypomethylated, and they give for genomic stability that is needed. And you have heard about jumping genes. So those transposons are located or they are regulated by these line sequences. And methylation, as I mentioned, that hypo and hypermethylation, that plays a role in that. Now in DNA methylation also 3 situations can come: abnormal increase or abnormal decrease or no change. And in increase, either methylation of both alleles or one allele and other mutated, or for imprinted genes, hypomethylated of one and mitotic recombination of one. And abnormal decrease like it was observed in proto-oncogenes or LTR. When gene therapy was developed at that time, retroviruses were used where LTR was considered like promoter. That time methylation was not developed, but in some recombinant experiment, this recombinant DNA did not work because methylation, that LTR itself got hypermethylated and got inactivated. So that's how it happened. When no change is there, then chemically induced mutations or poor repair of methylcytosine or deamination. So these are different combinations by which methylation regulates gene expression. But now we are in a situation that if we want to follow some pathway, especially by signal transduction which have been done, then we can identify which gene is getting methylated or which other epigenetic changes are there. Now, what kind of methods we use? So mostly for methylation, bisulfite sequencing is the one which can distinguish, this treatment can distinguish methylated versus non-methylated cytosine, and then you do regular sequences. Some are methylation specific PCR where you know the region and sequence in advance, and then you can make those primers and PCR-based technologies. But in all these methylation analysis, we keep track of methylation content, level, methylation pattern, profile, and pattern profile. And I will recommend you to read this review very nicely. They have described, Peter Laird, in this review about how to develop your control for methylation analysis. Steve Balin from Johns Hopkins, he has identified the sequence of other cofactors and other proteins when gene transcription is there. As you know, heterochromatin and euchromatin, they represent repression or activation, so that they have done. Similarly, polychrome repressor complex they have characterized. And one example is that if lysine 27 is trimethylated, then it is involved in differentiation. At the same time, S3K9, if this is monomethylated and next amino acid comes after lysine is serine, and then upon phosphorylation, that change becomes permanent. So those kinds of deep analysis for basic knowledge also are being done. Histone amino analysis is done by immunoprecipitation. It is called chip-on-chip or chromatin immunoprecipitation. So that analysis was done, and DNase 1 mapping is also done for that. And for that, basically what we do, that you have to shear the cells so that genome is disintegrated, and then use antibodies, and antibodies can bind to histone modifications, and then you can sequence the remaining one. And nowadays, ATAC sequence has also come where genome-wide profiling of chromatin regions you can do. So those technologies have come to us, and microRNA, those are relatively simple techniques since PCR and other things have come. MicroRNA, as you know, those are 21 nucleotide to 26 nucleotide, and they are very stable. So they are in any biospecimen, if you isolate those, like mRNA is unstable, but microRNAs are stable because of their secondary structure. So that also we utilize, and in that also 2 ways are there. MicroRNA information we use. First of all, those genes which code microRNA, that you can use as information, or microRNA profiling. And here biogenesis of microRNA are shown, but we are more focused on when mRNA and microRNA bind, and that profiling we take. So initially microRNA were in Drosophila or plants, but now we know in human, and not only in human, but cancer specific are also there. And microRNA gene coding polymorphism has also been identified. Now about 1500 microRNAs have been identified. And it is shown here that microRNA, depending on which site they are needed, they can change their function and they can move. So in the blood they can go, or through exosomes also they can go. And extracellular vesicles or exosomes, they are the ones where sometimes DNA, protein, or microRNA, they are there. From one organ to other organ they go. So they also provide a good biospecimen resource. About histone, I told you these are the octamers, and these are the modifications I have shown which in cancer they have been reported. And after that, in recent research you will hear about super enhancers or active enhancer, poised enhancer. They have been characterized based on different histone modifications. So if you want to go deep into that, those you can read about those. And about reader writers eraser I told, and histone 1 works as a DNA linker, and activating marks are S3K4 trimethylated, and silencing marks are S3 is histone, lysine 9 trimethylated, or lysine 27 trimethylated. They are almost universally present in most of the organ sites. Others are organ site specific modifications. And within that also such techniques are there that certain modifications you can follow temporarily or with different timings. So that study also you can conduct. And these are some of the modifications which either activate or repress, and whether they are mono, di, or trimethylated. So some modifications I am showing on this slide. Now single cell epigenomics is also coming. Mostly for basic research people are interested, so those analysis nowadays can be done in single cell also, and these are the samples which have been used, and it has implication in risk assessment or diagnosis or follow-up of treatment. So this article I wrote for the diagnosis of using histone modifications. And time to time we write some books or along with articles. So this I did for cancer epigenetics or cancer epidemiologies, emphasizing on epigenetics, one on biomarker, and last year I did on precision medicine, how we can use epigenetics in that. One time I was interviewed by Nature and they asked me that why epigenetics, or what are the features of epigenetics different than genetics. Then I mentioned that if genetic changes are there, you can at the most to follow that these changes are there, but you cannot do something about that. But epigenetic changes, you can reverse those by chemicals or some natural food. So those changes are reversible. So based on that, this was also another year for Nature. Then I told that some epigenetic modifiers or drugs have been developed, which are called epigenetic drugs. So that has epigenetics has slight edge over genetics. Whatever is done, like a lot of things are happening in epigenetics, at the same time, several databases are there, and most of them are such that people have access to those. Especially in NIH, if some funds are given to investigators, it is a condition that they should share data so that people can develop hypotheses. From different organs during development of cancer, exfoliated cells come, and they are also very good source. So if someone collects, then at different stages they can collect those cells and do methylation profiling. On this slide, I have shown for different cancers, a group of genes have been identified which definitely will be hypermethylated. And these are good biomarkers for that. And here, those epigenetic modifiers or those things are shown, and which genes are affected, they are also shown. So this number is increasing every year. You will see something more is coming. At the same time, I told you about the drugs. So different drugs are being developed. They are at different phases of development. SAHA is the one which is used also for patients. And again, these different epigenetic modifiers are there. Against that, these are the names of the drugs which are under development. So different pharmaceutical companies, they are doing. Like methyltransferase, I told you 3 kinds are there. HDAC or histone deacetylases are 10 or 11 kinds. So in different organ site, they are different. So people are making against that. And these agents are either short-chain fatty acids, aliphatic compounds, cyclic tetrapeptides, benzamide, those are there. So these HDAC inhibitors or epigenetic inhibitors, they provide a new kind of anti-cancer drugs, and they have been effective to stop cell cycle or in differentiation or migration or apoptosis. 2-3 examples I will give you, where in clinical trials these epigenetic inhibitors were used, especially in those people where other treatments were not working, so they were resistant. So azacitidine, which is for methylation, and this is for histone modification, when in combination they were given at a different time, pyrosequences and chip-on-chip was done, then they started giving response at day 1, 10, and 28, and on 55 people this was done. At the same time, combination of azacitidine, valproic acid, and oral trans-retinoic acid, they were also tried, and those patients who were not giving response to regular treatment, they started giving response. So these agents, they work like radiosensitizers and synergize the effect of regular cancer treatment. And these are also not only for leukemia or blood cancer, but other kinds of cancers, breast cancer, tumor, solid cancer. So different kinds of cancers, they have been tried, whether methylation inhibitors or histone inhibitors. And Schering-Plough or Bristol-Myers or Eli Lilly, all those are there. So idea is that those drugs, either alone or in combination, those should be considered. Now, FDA initially they approved 4 inhibitors: azacitidine, decitabine, valproic acid, and SAHA. But now list is increasing, so now 6 agents are such which are FDA approved. And these from the Nature reviews, or you can go to clinical trial sites at NCI. These are different kinds of clinical trials where these agents are being used. And you can read about those, which is the population, how much dose is given, and which one is given first, which one is given second. So all these I am giving as example, but you can go yourself and search. And if you type like clinical trials and epigenetic drugs, then they will appear. And sometime subtyping of cancer, like in breast cancer, maybe 50, 60 subtypings are there. In that also, epigenetic profiling helps. And if for different subtype, also different agents are working. So that information also we collect. That is very useful. And here also I have written for colorectal cancer, or combination of epigenetic therapy that was done in lung cancer. These are some of the publications. Similarly, in hematologic malignancy, that was effective. Now, a few examples now I will give for different type of cancer in which genes are epigenetic regulated. For example, in colorectal cancer, and at different stages, mismatch repair genes or APC, they are there. Now, CpG Ireland methylator phenotype, this was one phenomena which was for left side of colon. In that it was reported. Actually, left side of colon behaves differently than right side of colon. And in that, one side was found that which is sensitive to EGFR receptor, one was for BRF receptor, one side was CpG methylator phenotype or SIMP phenotype, we call that was there. So based on this analysis, that kind of treatment and other things were done. And nowadays we are supporting such research where microbiome is also affected in left and right colon, how we can utilize that information and integrate with epigenetic information. So here, based on methylation profiling, KRAS in red, BRF in BRAF mutation in green, and microsatellite instability, that high or low, all those were integrated, and those people who were at high or low risk, those could be identified based on this. So this kind of analysis helps us in identify high risk population. In AML and ALL, just because of based on methylation profiling, AML and ALL with healthy, they could be distinguished. In lung cancer, these are the genes which are affected, but along with that, some proteins were also identified. So integration of proteomic structure, proteomic information with epigenomic information was done. And these are the genes which are regulated by methylation in lung cancer. And lung cancer especially, since sputum is the easily accessible sample, so that has been characterized nowadays. Asbestos exposure, that was contributing to mesothelioma. In that also, methylation profiling helped us. In Barrett esophagus, which is for esophageal cancer precursor, in that for 20 genes were taken. Some genes get methylated at early stages, some at later stages, but in the same patient, several times samples were taken and those markers were identified. So that was correlated with survival also, methylation profiling. In pancreatic cancer, why we take patient-related information? So suppose someone is diabetic, alcoholic, and P16, KRAS mutation is there, all those features are there. Along with that, P15 and P16 are methylated, then that person is at high risk of pancreatic cancer. Otherwise, this cancer is such that by the time you diagnose, it is quite late. So in that, epigenetics has helped. In breast cancer, we can follow response of treatment also based on this methylation profiling. And also that has helped us recently. Many times people do over treatment. So how to distinguish aggressive cancer with indolent cancer? In that also profiling of epigenetics, that helps. Now in cancer, infectious agents, about in 15% of cancer, infectious agents are involved, and some of them are regulated by epigenetics, in which latency-associated nuclear antigens or Epstein-Barr virus nuclear antigens, those have been identified. And complete methylome of papillomavirus, which is for cervical cancer, or Epstein-Barr virus, nasopharyngeal carcinoma, or HBV for liver cancer, that has been done. Why we do that? That before any other symptom, if you have profiling, then you can identify high-risk population. And in that also, like early genes or late genes, which are mostly in viruses, how much methylation is there, which site, and when it will appear, that we can do. And distinction of SYN1, SYN2, SYN3, which are different stages of disease development, that can be done. So based on methylation, you can triage also different women which are at high risk of developing cancer. So that can distinguish SYN2 with SYN3 different stages. And then it was also observed that elevated methylation of HPV, L1 and L2 open reading frames that distinguish invasive cancer with normally developing cancer. And this kind of article we wrote for HIV, AIDS, and AIDS-related malignancies like Kaposi sarcoma, HHV-8. If in AIDS patient it infects, then Kaposi sarcoma develop. And in liver cancer, hepatitis B, hepatitis C, all those viruses contribute, so that is also infectious agents regulated. And sometimes these kinds of observation we see like in Japanese population, SOC, SOSC1. This gene was the one only in Japanese population that was methylated, not in others. So for oral cancer also, methylation profiling genes or group of genes have been identified. In immune system also, T cells and B cells, by different pathways they have followed and found in different papers it is reported that epigenetic regulation occurs. So inflammation, as you know, that is a common factor for most of the cancer. So whether it is for T cells or B cells, that has been done. GSTP1. This marker has come for prostate cancer. So PSA is the most common one. But if you follow GSTP1 methylation, then sensitivity and specificity, that increases. And based on that, Epigenomics, one company, and Quest Diagnostics, they have taken charge, or they are the owner of this test of GSTP1 methylation, which is being done and it is in clinic. In bladder cancer, exfoliated cells you can use to follow methylation profiling in urine, and such techniques are also there. Sometime when cancer develops in one organ site, then in other sites also it might develop, called secondary cancer. So at a time, what is happening in terms of methylation profiling in other site, simultaneously you can do, and Peter Laird of University of Southern California, he has developed those technologies. Now comes to its prevention. Like in cancer prevention, they say that 40% of cancer by changing lifestyle or diet, you can do that. So those kinds of evidence are coming. And from natural food or bioactive food components, those agents are being identified which has that kind of role or significance. And I also wrote one article on cancer control and prevention, nutrition and epigenetics. And in that, certain foods were such where epigenetic inhibiting properties were found, whether against methylation or deacetylation. Those were there. And since we know about many of those genes, their chromosomal location, so we can take advantage of that. In liver cancer, just by changing methyl donor, that kind of models were developed, which were where no external carcinogen was added, but that liver cancer could be followed. And by nutritional supplement in liver cancer, much progress was followed in that case. And this is very important slide because these are such food and food components which generally people take. So some of them have DNA methylation inhibition properties and some have histone inhibiting properties. So Institute of Alternative Medicine and some other investigator, they are exploring that idea also. And here the structure or the chemical structure in those bioactive food component, those in different kind of references, if you want to read more about that, that has been described a lot. So what kind of research opportunities these research provide us and what challenges are there? So few questions are such like, if we include epigenetic markers, whether we can identify new risk factor or not, whether they are associated with different races or ethnic groups or not. In US, as you know, people come from all countries, and some drugs will work in one group and some work on another group. In that, whether that can help or not. In epidemiological studies, how we can use that to increase sensitivity and specificity. And genetic and epigenetics, how they are correlated, whether race or ethnicity specific microRNAs or non-coding RNA are there. How can we use information from, by defining subcategories using epigenetic information, how we can overcome technical challenges in EVAS studies, or can we predict recurrence or secondary cancers, how efficiently we can do that, or how we can harmonize epigenetic data. When data comes from different sources, so the way they collect, the way they store, and the way they have used their biospecimen, how we can integrate that. What is the window of susceptibility when people are more sensitive? And since these epigenetic phenomena are in all cells, so whether inhibitors, how we can make those specifics for some specific cells. How non-histone proteins regulate, and cancer stem cells, that is another topic. Like in cells, they say that 1 out of 1 million cells will be cancer cells, how we can characterize more so that we can target those, or how we can utilize microbiome specific information for control and prevention. So how we are addressing those changes? So in few slides I will show what at NCI level, at NIH level we are doing. That will tell you the status and challenges. So roadmap program was initiated from NIH OD, in which all institutes participate and about 1.2% of NIH budget goes. So in that, epigenetics was one of those programs that was selected because it is involved in several diseases. And this program provides such money, like these are multi-million-dollar projects, so that such projects can be done the way human genome sequencing was done. Similarly, we wanted that in healthy people, human epigenome sequence can be done. So that was the objective, to develop technology as well as doing epigenome map of human. And these are the programs which are supported by, initially it used to be called Roadmap, now it is called Common Fund. And per year about $500 million we do. So epigenomics is here, 4D nucleome I mentioned, microbiome or extracellular RNA. So different kinds of programs are there. In this program, since its conception or initiation, I have been representing NCI, like metabolomics, epigenomics on molecular transducers of physical activity. And they update by one newsletter, it's called Common Fund Connector. For epigenomic, as you see, this is the total amount we dedicated for a few years, and then epigenomic maps were developed, and Nature gave full volume dedicated to epigenomic roadmap. And in this way, since all cells have different epigenomes, so 111 kinds of cells were isolated. These are those, and you can read about that. And then in that, epigenomic methylation, microRNA, and DNase-1 mapping was done. In that program, several papers were published in Cell, Nature, Science, otherwise also. And one paper which was genome-wide methylation analysis that has been cited about 2,000 times. So high quality of papers came. At international level also, one program called IHAC, or International Human Epigenome Consortia, was developed. And in that, idea was to complete 1,000 epigenome from different regions. And in that, in initial meeting, I participated, and I represent human epigenome of NIH. But these are the countries who participated in that, and different agencies are funding that. And industries like Affymetrix or Novartis, they are also participating. And Nature and Science, they covered about IHAC program, and AACR, which is American Association of Cancer Research, that society also covers. So this is the website for IHAC. The difference in roadmap and this one, that they are doing animal studies also along with human. Because many things, we have to use animals to understand what is going on. So now we have several partners, and through ENCODE program or dbGaP or other, all data is being stored, which is shared among all. And for that also, we write time to time how we can, we discuss about challenges or opportunity in these places by writing articles. Like for personalized medicine, since we know the profiling, and we know that different people respond differently to treatment and intervention, so those things help. So this I did for personalized medicine. This program is also one common fund program. It's called MotorPack, or Molecular Transducers of Physical Activity. As such, physical activity we know, like in lifestyle, that helps in disease prevention. But here we want to know what is happening to these transcriptomic analysis or metabolome or genome or other components, whether they help or not. So it is designed in such a way that for 12 weeks, a group of people will be doing definite exercise and one group will not do. And before that and after that, several muscle sample or blood samples will be taken, and metabolomics and transcriptomics and epigenomics and genomics analysis will be done, and that will be integrated. So that just 2 years ago we started, but this is also such, like $30–40 million per year for 6 years we have been involved. About ECHO program, environmental influence on child health outcome, I mentioned that here focus is on children before birth or just after birth. That also will help us in understanding that. AACR has developed one data sharing program. It's called Gening, because data different people do and store, how we can utilize that. And at NIH, personalized medicine program came, brain program came, then cancer moonshot program came. So these are also different programs, like our Francis Collins initiated precision medicine, in which 1 million people will be enrolled and followed for different molecular analysis and subtyping. And now its name has been changed from precision medicine to All of Us. So it is called All of Us. That was also started. This is also congressionally mandated. So you'll follow how this thing works. But these are the ones where most of the knowledge which we are gaining through basic science and all these programs which I'm telling, how to integrate that, how to utilize that to identify population at high risk and developing prevention and treatment strategies. So in this also precision medicine, they are emphasizing that we should use this profiling for identifying patients or developing these technologies. Implication. Cancer moonshot program is such, Joe Biden started that, and his idea was, because as you know, his son died when he was 41 years old. So he also put some, asked NIH and Congress to put dedicated money to NHGRI, which is Genomic Institute, and NCI Institute. And idea was that whatever we are doing in terms of treatment in 10 years, can we do in 5 years if we increase the money? So about $200 to $300 million were assigned, and these were the objectives. So whatever is the existing technology or knowledge, how we can use that maximum. So that has started. This is called Moonshot program. For that we have Blue Ribbon panel, which are the people expert in the cancer area, and they give recommendation, they follow the progress also. So for personalized medicine, this article I wrote, or challenges in epidemiology or genomics research at NCI, or this was for stem cells, that also I have done. And these I am giving because any of the chapters or article, if you want to read later on your own, that you can do. And in those we write what is the future, what we think about that, why NCI is putting some money in some program and not in others. So those justifications are there. This was also a very good program. It's called TCGA, or Cancer Genomic Atlas. Very well identified tissue samples from patients were taken, cancer patients, and profiling was done, genomic, methylation, and others, and that database is there so that people can use for their research. And now 2 years ago we have started PCGA. It is called Pre-Cancer Genomic Atlas. So those will be reference maps which people can utilize later on. And integration of all these omics, that is the key to success in future. That message we do. These are some people who have helped me in doing all these kinds of projects. And collaboration, that is the success for our things. And now I will take any questions that you might have.
Perhaps you could comment on how DNA methylation patterns and histone acetylation patterns change during carcinogenesis.
So for that, sequentially collected sample should be there in which we follow both, and that tells. So initially what people were doing, that they have collected samples, at the end one time they have measured. So whatever is happening, but if you want to know when it is changing, when it is more, or whether one is expressing or other is expressing, that can be followed. And in animal studies it was done first, and now in human, samples are being collected and profiling is done. And specific genes have also been identified which in certain diseases they contribute. So when we go deep into some site, a specific site, like in colon cancer, maximum studies have been done. So in that it has been characterized much more. And some of the data which I was mentioning about early stage, or in human roadmap also, when mapping was done, then stem cells, embryonic stem cells or induced stem cells were taken, and they are from 2 to 8, 8 to 16, 16 to 64. In that profiling was done. And then also it was seen whether histone came first or methylation first, and which modifications were there. These are difficult experiments, but that's how it's happening. Yes.
How much the methylome is, or the epigenetics is organ specific or cell type specific, and how much is
whole gene? Yeah. So whole is mainly in line sequences, those I told, and those are about 70 to 80%. So only 20-30% is gene specific or organ specific, because in organs are gene, and within that also mostly it is in promoter, but sometimes you find in other places, or enhancer also. For example, in genome-wide association studies, one side 8q24 was identified, and that locus was for colorectal cancer, breast cancer, prostate, and many. And then 250 kb far was MYC gene, but enhancer, because of chromatin modification, that was taking a fold, and that enhancer was coming to the Tata box, and that's how it was expressing. But your answer is that is a little portion. Yeah. Yes. Yeah, those like 6 out of 6, I think 4 are demethylating agents. And I gave the examples of those clinical trials and others. So those are so much effective in those. Yes. So examples like 84 trials, etc. I mentioned, and many of those sites. And on your own also, in Google you put clinical trials or success of epigenetic inhibitors and all that. So those clinical trials will come. Mostly epigenetic drugs are working in combination. Like regular anti-cancer drug you use, that will be less effective. But if you use like PARP inhibitor, SDAC inhibitor along with that, then suddenly it starts giving that. Yeah. Okay. Yes. Yeah. So the reason we call it atlas and the way it is made, first of all, they are very well characterized. When tissue is taken, that was carefully taken. Analysis was done. Suppose some patient come, you have some sample, and people are analyzing by profiling. So in those cancer, at least they can match with something, that if this sign is there, this profiling is there, that belongs to this cancer or other cancer. So that will be a reference map. And similarly, imaging maps are also being done. That has nothing to do with this methylation, but by imaging also. Previously, everybody will depend on pathologist, how much expertise. If pathologist says, that is the way we have to do treatment. But before pathological symptoms come, much earlier disease develops. So these things will help there.
We'll have to move on. Thank you.
Yeah, thank you.
Our speaker for today is Frank Maldarelli. He's with the HIV Dynamics and Replication Program. And he got his Ph.D. at City University of New York, M.D. from Mount Sinai School of Medicine. He did a residency in internal medicine and then came to NIH, first with NIAID as a medical staff fellow, and then he joined the HIV drug resistance program. And he does extensive collaboration. Mostly he's up at Frederick, and he collaborates with the NCI HIV and AIDS malignancy branch and the NIAID AIDS clinical research program. He's currently a member of the NIH Infectious Disease Advisory Board, and he's a faculty member of the International Society of Infectious Diseases HIV training program. And he's going to tell us all about HIV today. Frank.
Thanks a lot, Terry. And thanks for the opportunity. And hi, Frederick. They're listening in Frederick. Is that the idea? Yeah. All right. So I see there's a huge crowd. If you have any questions, just ask them. This can be a very interactive session. And I think that, you know, the title of this course is TRACO. May belie some things that say, well, what are we doing with the retroviruses lecture? And I hope to at least address that issue, but realize that even long before HIV, there was a huge program in retroviruses in the NCI. And so this is a long-standing interest because these viruses don't just cause immunodeficiency, they also cause cancer. And so what I'd like to do is just by way of introduction, talk about the process of retroviruses and where they come from, perhaps, what their molecular biology and replication program is, talk about the retroviruses in human populations, and then how they emerged, how they spread, HIV therapy and beyond, and then perhaps some lessons that at least branch out into other areas of interest. So if we look at this just, and I'll get to some other retroviruses, but just from the standpoint of HIV, the point I'd just like to make is that in this day and age, there are close to 37 million individuals infected with HIV throughout the world, most of them concentrated below the equator. So the highest amounts in Southern Africa, 19 million, about half. But then large populations here in the Southeast Asia and Latin America. Above the equator, somewhat less. This doesn't tell the whole story. I think one thing we'd always like to know is where is it changing most quickly? So this is the prevalence of HIV on the planet. What's the incidence like? And so areas such as sub-Saharan Africa have a much more stable epidemic, something people would call endemic, where the same numbers of people have been infected over long periods, whereas places like Eastern Europe, the numbers are increasing much more than in other places. So they both illustrate the problem, but some places at this point are worse than others, in terms of trying to stop transmissions. All right, so I got to ask, right, since there are so many people here, if you had a guess, how many people of this 37 million are now on antiretroviral therapy? And if you're in Frederick, just shout it out. We can hear everything. Half. Other guesses? Quarter? You think closer to a quarter? You're nodding. And about, and here, any other guesses? So that's a worldwide number. Okay, I got to ask another number. How many people do you think in the United States are infected? All right, hold that one. Are on therapy. Okay, so total, about half is absolutely right. Did you look at these slides? I'm just asking, okay. But that really is a huge development over the last 5 to 10 years, and it's really, I think, the work of many countries and many institutions. One of them being the NIH, and I would put this on Anthony Fauci, because a number of years ago now, and I really leave it to him. I think he convinced all the right people that having a president's emergency fund was a good idea. So PEPFAR, one of the things that was a driving force to bring antiretrovirals around the world was the work of people here at NIH convincing all the right people. So about half. In the United States, right, the need is still great, and HIV is a challenge on numerous levels. So the prevalence of HIV in the United States is actually increasing. Number of individuals per country is going up. And that's because the number of new cases of HIV is really, it's gone down in the last couple of years, but it's relatively stable at about 38,000. Because of antiretroviral therapy, the total number of deaths has gone down.
How does the opioid crisis affect
Like a lot. Let me come to it. Let's see if I have it on this slide or later. This, I think, is, if you had to remember one thing, you got to remember one thing. It might be this graph, and it's not one I put together, but it's something called the Gardner State of Engagement in HIV Care. And it really tells us what the state of the epidemic is in the United States, because it doesn't tell us everything. So the first column here, bar, how many people are infected? Estimated. And that's somewhere around 1.1 million. How many people are actually diagnosed? 900,000. Based on that 900,000, what people say is there must be more out there that we don't see. And they use a complicated algorithm to estimate that, well, if we captured 900,000, then there must be this many more. So at each level, it tells us where our challenges are and where our opportunities are. If this is all we have, then we have to get to these. If we're using standard passive surveillance, people come in for routine care and get a test, that's passive surveillance, and we've gotten that many. And what we really got to do is perhaps use an active process to get the rest. In other words, go out and find populations that are not coming into care. I'm going to come back to your question when I get to how many of those people are linked into care. Of this 800,000, it's down around, I don't know, 600,000 or so. How many people who are linked into care are actually retained? Somewhere around 400,000. How many of those 400,000 need therapy? The same number. Essentially everybody should be on therapy. And how many of those are actually on therapy? Somewhere around 300,000 or so. Of those who are on therapy, how many are actually adherent? Somewhat less of that, maybe 80%, and essentially less than detectable. So coming back to your question, right? that opioid crisis is greater or less in different places in the country. We found out in the last 2 years that Appalachia is a place where it's turning out to be quite bad. And there was a remarkable epidemic, perhaps it's the one you're referring to, 180 or 200 individuals in a town that probably didn't have more than a few thousand people in it who became HIV infected, linked to 1 and maybe 2 point sources. How many more of those are there? And the answer is, but we found a few more. Does that tell us this number needs to be addressed in a different way than simple passive surveillance? Because individuals were going to get their opioids from wherever and still not being tested. So knowing that everybody in the United States, if they go to a routine physician's visit and have the ACA or Obamacare, they can be tested every year. These individuals obviously should have been tested more so. So what numbers are those? Those are in the hundreds to low thousands. Every year we get about 38,000, and I think it's maybe on the next, perhaps not. Most of those, 60 to 70%, the risk is men who have sex with men, and I don't know yet, and nobody does, what the opioid epidemic is going to do to this number. But if you had a guess, it was probably at least a few percent of the total. And whether that goes up or not is going to be a real issue. So if we have fewer deaths because we have this many people on antiretroviral therapy, right? So people are not dying the way they were 20 or 30 years ago. If we have, thankfully, many fewer, 6,400 deaths, then the total number of individuals with HIV infection is going to increase. Because of that change in prevalence and the effect of antiretroviral therapy, there are now distinct areas of research. So if you look globally at 30,000 feet, this is how the NIH spends money on HIV. And it's funneled through the Office of AIDS Research, comes right out of the NIH and the HHS budget. They've defined four simple areas for research: reducing the incidence of HIV, next generation HIV therapies, research toward a cure, and HIV comorbidities, co-infections, and complications. So obviously you can see some areas of common ground for research, but this is the list of things that the Office of AIDS Research is concentrating on. Or to look at it another way, if you want to get a grant to do HIV research, it's going to be in one of these areas. And engaging as much of these areas as possible is probably going to give you a better score on your grant. So much of our work in the Dynamics and Replication program is here in research toward a cure. But because some of the things we do have direct relevance and application to reduce the incidence of HIV, we use techniques that we've developed here to study how HIV spreads in populations. And just having the kinds of techniques and optimizing them helps us understand how new outbreaks occur and how to model them. So these specific goals, if you're interested in HIV research, take a good look at this and see where you fit. The only way to get to any of those goals is to understand the molecular biology and the replication of HIV. And that's what I want to talk about. That's probably it's probably the most important part. You can't derive the entire epidemic from the replication program, but you can get a lot of it. And so if we were going to define a retrovirus, it's a group of RNA viruses that replicate via DNA intermediate using reverse transcriptase. And so when that sentence was finally defined, and I would say that would be in the late 1960s, early 1970s, that became a paradigm shifting sentence. And that's how it got its name. Is this a surprise to you all, retroviruses? So in the 1950s, okay, they found like DNA, and it was like the genetic material. And between the 1950s and the early 1960s, largely through some work done here by Marshall Nirenberg, we found that, yeah, we have DNA, it makes RNA, RNA makes protein. That's the paradigm, right? We have genetic material in DNA, it makes a messenger RNA, that messenger RNA makes protein. DNA to RNA to protein. Now along comes a retrovirus, and the virion itself is an RNA virus. All right, so okay, we have other RNA viruses. RNA viruses have special replication enzymes. RNA makes RNA, that RNA makes protein. Simple, except for this. In this case, the RNA that's in the virus gets made into a DNA copy. That DNA copy gets integrated into the host genome. And now, because the enemy is within, it makes RNA as a function of whatever else the cell does. So RNA went to DNA, went to RNA, went to protein, went to virion. So it was going in reverse. And that's how it gets its name as retrovirus. The enzyme central, two enzymes that are central to its replication, one of them is called reverse transcriptase. It takes RNA and makes a DNA copy. Up until that point, everything had a DNA and it made an RNA copy. So they called it a reverse transcriptase, right? That's how it got its name. But this entire paradigm, this entire set of viruses shifted this paradigm that says DNA goes to RNA, goes to protein. In this case, RNA goes to DNA, goes to RNA, goes to protein. So there was a critical missing enzyme, reverse transcriptase. And then people found that it might actually answer another challenge. It's not just a different paradigm for replication, but if you think about it, what are the oldest enzymes? They're not proteins, they're RNAs, right? And it's a theory, but people think that we used to have an RNA world. The atmosphere was reducing. And the thing that was replicating was an RNA genome. That RNA genome was replicating its own RNA genome, and it's a theory, and DNA really had nothing much to do with it. Unfortunately, the organisms that were RNA in this RNA world, the organisms were making a byproduct. Their poop was oxygen. And if you've ever worked with RNA at the bench, anybody worked with you open that tube, you're already losing it because oxygen it's one of those things that make an oxygen radical, and it can destroy the sugar base of the RNA. DNA is deoxyribose, and it's stable. So as oxygen accumulated, these organisms were sort of in trouble. And so the ones that survived were ones that could store their genetic material in something that was going to be stable to oxygen. That's the hypothesis. And therefore, DNA. And therefore, the organism, before you get into any viruses, they all have a reverse transcriptase in them. And so one theory is that that's the reverse transcriptase. That reverse transcriptase essentially changed the way life evolved on the planet. Some people would say it saved life on Earth, or was it Jedi Knight that destroyed the possibility that oxygen was going to get rid of us. But if you look and take a walk around, I know you guys are never allowed out, right? This is the NIH. But if you were ever allowed out, this is Muddy Branch up in Great Seneca, up off Clopper Road. Anything you see that's alive has got a reverse transcriptase and a retro element. Every last one of them. The birds, the plants, the fruit flies that are in my kitchen, the roundworms, bacteria, retroplasmids, the fish. All right, so we don't have any pigs. You got to go to the store for those. The rodents and the primates all have this retrovirus. That all have this reverse transcriptase. When it comes down to retroviruses, you can classify all the retroviruses based on the genetic relatedness of that reverse transcriptase. And you can divide these viruses up into about 7 different types that pretty much cover the so-called waterfront. And the reason, and this is sort of a family tree, you've seen your family trees. Usually give this lecture after Thanksgiving, so it's a little bit easier. All right, everybody who showed up around your table, you could probably design a family tree around it. Here it is for retroviruses. The thing I want to point out is in bold are the retroviruses that are found in humans. And so here's one down here called human T-cell leukemia virus. Here's another group, lentiviruses. This is what HIV-1 and 2 belong to, as well as a bunch of others. And then up here, spumaviruses. It's an exogenous infection of primates. Well, you know, it's kind of interesting that they don't all belong, or they don't all fall on the same branch. They're distinct viruses, all of which infect humans, but none of which are related to each other. Spumaviruses are most closely related to a virus in mice. Lentiviruses in humans, HIV-1 and HIV, their most close relatives are in the chimpanzee and in monkeys. And for the, you know, after you get beyond them, there are viruses in horses, in goats, and in sheep that are more related to HIV than they're ever related to this spumavirus. And HTLV, don't get me started. It's related to viruses that are in cows, right? And maybe closely related to some that are in chickens or in mice or perhaps in some primates, but more distantly related to lentiviruses. So the viruses, the retroviruses that infect humans are not all, yeah, they're all related to each other in general, but they're not close relatives. The HIV-1 group and HIV-2, they're relatively close, as we'll see, but in the grand scheme of things, there are 3 groups of viruses and they're not all related to each other. When we cone in now on those lentiviruses, we'll see the same thing. Here's HIV-1, that infects 36 of the 37 million people in the world infected with the human immunodeficiency virus. Its closest relative is in the animal kingdom in chimpanzees. HIV-2, about 1 million people in the world. Its closest relative is not HIV-1. Its closest relative isn't even in a chimp. It's in a monkey, a sooty mangabey. And so right away, this tells us a couple of things. First of all, we can get hit on a number of sides. There are, I'm sure you all know this, about 144, 145 different species of primates that are not humans, and about 44 of them, about a third, have relatives of HIV of one form or another. 2 of them have made it into the human population, probably, most likely, in distinct introductions. And in fact, perhaps this HIV-2 has been introduced a number of times into the human population, and the HIV-1 has been introduced a number of times, a number of different groups. So you might think of it as the human population being assaulted at different times with viruses in the primate kingdom. Here's, in terms of conventions, this is what the virion genome looks like. The virion RNA, when it was mapped, looks different from the DNA when it's integrated into the host genome. So this became a molecular biologist's fantastic puzzle. How do you start with something like this and end up, and we'll see what it ends up as. But the first thing they noticed is that there was a region at the 5' end and a region at the 3' end that were identical. They named them R for repeat. Those were followed by regions in the 5' end and the 3' end that didn't have coding sequences, but they were unique. So they were called U5 and U3, the unique region in the 5' end and the unique region in the 3' end. By now you should get the sense that retrovirologists are very concrete people, right? R is for repeat, U is for unique. These genes that followed open reading frames had coding sequences in them. And when you looked at similarities between the sequences, you could find promoter-like sequences, but they were at the 3' end. and you could find a polyadenylation signal, but it was at the 5'. So like, what's up here? Okay, tell you in a minute, but we labeled the rest of it according to, as you'll see, different conventions, but primarily polymerase genes, envelope genes sit on the surface, and GAG group antigens, as well as a number of other genes. That's what it looks like in the virion. Here's what it looks like in the cell below here. Now, once it gets integrated into the host genome, that unique 3' end region is now at the 5' end. And the unique 5' region is the 3' end. So like, what's up with that? What's up with that is they sequence the virus before they sequence the provirus, what's inside the cell. So somewhere from going from here to here, this U3 made its way up here, this U5 got duplicated at the end. This whole region is now a long terminal and a complete repeat. So it's called an LTR, long terminal repeat, because this region is now identical to this region. And now, for what it's worth, I got promoter elements over here, and I have a polyadenylation signal, the second one, over here. But if I suppress this one, I only use that one. This is how it makes its RNA, using this as a promoter, +1 for the RNA here, and all of the genes follow. The protein gene products are capitalized. The names of the genes are in lowercase and italics. Here's a glossary of terms, names all the genes, GAG, not because this is making me gag. By now, I'm getting sick, is really for group antigen, a serologic term that distinguished all the viruses before we could sequence them. Pol for the enzyme that makes the polymerase, N for envelope, a protein that's going to stick out on the surface of the envelope protein, of the envelope virion, TAT and REV, things that are genetic activators, one that transactivates transcription, and a second one that regulates spliced messages in the cytoplasm. And then here, a list of all the other RNA sequences. If just looking at it from the virion standpoint, here's a artist depiction of that. On the outside, a lipid envelope that it gets as it's budding through the membrane. Viral glycoprotein sitting on the surface, and then on the inside a core. That core contains 2 RNA molecules not complementary to each other, 2 identical RNA molecules, and then as well the enzymes it needs for reverse transcription, for integration, and then for protease. So here, let's just go through the sequence of what is going on here. Well, the first thing that goes on here is that the virion has to attach, it has to get into the cell. And getting into the cell, you'd think, might be like, okay, it's got a lipid envelope, cell has a lipid envelope, the 2 of those things stick together, and the next thing you know, they've fused and it's fine. It's like bubbles. You know, you see bubbles floating around, you'll see 2 of them get together. Okay, well, that'd be nice, but that isn't the way it happens. Bubbles are unilamellar membranes. Remember, these are bilayers. They're not just bilayers of lipids, they have protein stuff in them. To get this virion through this membrane takes a lot of energy. All right, to get this virion to this cell takes some specificity. So I need a system that gives me both specificity and enough energy to push it through that membrane. And that's where we have distinct functions for the HIV surface protein, GP120 and GP41. So to get in, we have to attach, and then we have to enter or fuse. The viral factors for attachment, GP120, the viral factor for entry, GP41. GP stands for glycoprotein. The number stands for the apparent molecular size in an SDS gel when you run these proteins. So it's got about a molecular size of 120,000 Daltons. This one's about 41,000 Daltons, and it's a glycoprotein. All right, That's what the virus is bringing. What is the host bringing? A receptor, CD4, but that's not enough. You need a co-receptor as well. So this protein has to bind to both CD4 and a major co-receptor, either a surface molecule called CXCR4 or CCR5. Working that out, molecular biologist's dream, right? But working that out gives you a place to stand and develop inhibitors. And one of them, most recently developed and FDA approved, is a drug, antibody, called ibalizumab. And that what that does is blocks the entry of the virus by blocking the surface, blocking the virus binding to its surface membrane, the surface CD4. By being able to block that, we prevent attachment and prevent fusion. That's the interaction between GP120 and CD4. There's a second interaction that it needs to make to the co-receptor, and we have a second drug called Maraviroc that blocks that interaction. So one blocks to the receptor, one blocks to the co-receptor. The receptor one is so new that we don't know how well it works with the others, but we don't know whether it's totally additive or synergistic with a second drug. But they don't interfere with each other. So we have two ways to block just the virion getting to the cell surface. Well, as I mentioned, we have to get inside the cell. Attachment's not good enough. The fusion reaction is mediated by GP41. So upon attachment to the receptor and the co-receptor, there's a rearrangement in GP41, which is non-covalently bound to GP120. It rearranges itself into 6 helix bundles. And now those 6 helices have in them the potential energy to push the virion through the membrane. And you can just imagine, you guys know alpha helices. There's plenty of potential energy there, and that's what drives the virion through the cell membrane. Once that's done, then the virion plops itself into the cytoplasm. Well, knowing that interaction and knowing that it required these alpha helices, what the molecular biologists developed was a drug that would essentially be a monkey wrench thrown into those alpha helices. I'm sorry, that's a drug called enfuvertide. It binds to these bundles, these 6 helix bundles, and prevents fusion. So before the virus even gets inside the cell, there are 3 ways to block that. All right, so let's say it gets past all of those because we didn't use those drugs. The next thing it has to do is now the virion core is in the cytoplasm, and it has to undergo reverse transcription. And here in this, we've gotten through attachment and fusion. Now it has to uncode. And that process of uncoding is terribly poorly understood. And it's a shame, really, because if we knew how encoding occurred and we could block that, we'd probably have a very, very potent inhibitor. And the reason is simple, that we think that that would be a very potent inhibitor, because we know that there's a blocker in the cell now that blocks incoming viruses. It's called TRIM5 alpha. TRIM5 alpha is a sensor in the cytoplasm that finds, it recognizes patterns. It sees the core. And when it sees the core, it can block infection. Well, that's terrific, except it doesn't do it for us. It does it for a lot of other viruses, and TRIM5 alpha in other species blocks HIV. And what I mean is described on this graph, this table. So everybody's got a host innate immune protein called TRIM5 alpha. We have them, chimps got one, monkeys have them, fine. Everybody's got one. Well, the human one cannot block HIV. The chimp one cannot block HIV. The monkey, TRIM5 alpha, completely blocks the ability to HIV to get in. So you can get it to attach, you can get it to fuse, you can get it to come in, and it doesn't replicate. And the reason is its TRIM5 alpha blocks reverse transcription of HIV. Well, you know, we're pretty close relatives. What's the matter with our TRIM5 alpha? And it turns out what's the matter with our TRIM5 alpha is probably can be summarized in just 1 or 2 amino acids. That's how many differences we have. If we change our TRIM5 alpha to the monkey one, we can block HIV infection completely. It's that good. You know, for the lack of a nail, we lost the war? Pretty much. Why is it? Why did we get so unlucky? Well, take a look at this. The chimp, TRIM5 alpha, the chimp virus can infect humans. The chimp virus can infect chimps. Chimp virus does not infect monkeys all that well. All right, well, how about the monkeys? They get viruses too, right? Well, their monkey virus can infect humans. It infects chimps, but not so well. And it infects monkeys. So there's something different about their virus, the monkey virus, that allows it to infect monkey cells and human cells. How did we get so unlucky? Well, as it turns out, TRIM5 alpha turns out to be a very quickly changing, it's probably one of the fastest evolving genes in primates. So if you line them all up, you know how this goes, right? You line up beta globin, there's a couple of changes. You line up another gene, there's a couple of changes. You line up TRIM5 alpha. And for monkeys to humans, it's quite a, and you go through the primate lineage, you can see that there are an enormous number of changes. Not a lot between the final monkeys and us, but enormous number over time. And so one working hypothesis is that it's changing to accommodate waves of these viruses that are infecting these populations. In other words, our TRIM5 alpha, yeah, it's just a couple off, but it saved us from the last infection, the last wave of viruses that came through. The last one came through and maybe destroyed a whole swath of humans, but the ones that had the TRIM5 alpha we have now were able to resist that virus infection. So we're the success of the last wave. And this is Charles de Gaulle, who said, generals are always fighting the last war. We won. We are the winners of the last war, but we're still fighting as if it is the last war, when in fact, what we need is a TRIM5 alpha that can resist HIV infection. It's possible it's out there. You know, there's a variant in a human population. And if we didn't have antiretrovirals, those might be the people who survive. So evolution can solve this problem, but it's going to take too much time. And so, you know, we live in a much faster society. And so we do things differently. All right, so once it gets past this uncoding step, reverse transcription takes place. These are the wonderful steps of reverse transcription that show us how you can get from that RNA genome with just an RNA U5 and a U3 and an R to a DNA proviral genome that has both. It requires molecular gymnastics that we don't need to go through today. But if you want to look at it, it's sort of cool. The one piece of information that is useful is that everything on the planet, right, that's going to replicate needs a template, a primer, and an enzyme. The template we know, that's the RNA. The enzyme we know, it's reverse transcriptase. Where's the primer from? And it turns out the primer, it was stolen from the last cell I was in, and it's a tRNA. So the tRNA, and in the case of HIV, it's for lysine. tRNA lysine binds to the RNA on its way out, and that's how it starts its priming, its replication. All right, so it makes its DNA copy. That DNA copy then either happens at the nuclear membrane or sometimes in the cytoplasm or sometime in the nucleus. Sooner or later, that DNA copy is in the nucleus and it has to be integrated into the host genome. That's mediated by a gene product called integrase. It makes a cut in the host genome and then integrates the host genome. At that point, as I say, the enemy is within, and then it's 10,000 nucleotides. It gets treated like anything else in the host genome. It's got a bunch of promoters that are recognized in a lymphocyte. It makes RNA through RNA polymerase. It doesn't bring its own. It gets transcribed in that fashion. The RNA is then transported out of the nucleus, again, with a cooperation between the host and the virus, and then gets translated on totally cellular ribosomes. I get ahead of myself. These are the enzymatic activities of HIV reverse transcriptase. It can make DNA out of an RNA. It can make DNA out of a DNA because it's got to make both strands. It can get rid of the RNA and the RNA-DNA hybrid. And it can make RNA. Here's an RNA-containing virus with an enzyme with a lot of faith. It doesn't make any RNA. But it does make mistakes. Anything that uses RNA as a template always makes and has an error rate that's much greater than a DNA template. In general, it's about 1 mistake to 4 mistakes out of 100,000 bases synthesized. The virus is about 10,000 bases. Bottom line is, once you get through the first round and the second round, about every time that makes a virus, it makes a mistake. And you can get recombination during the reverse transcription events, permitting a reassortment of all kinds of sequences in the genome. Replication is rapid and error prone. So you can just imagine, how does this thing ever make anything that works? If it's making that many mistakes and it's replicating so quickly, it sounds like this is some leftover virus. But what it really means is that this is a pathogenic determinant for this virus replication. The fact that, yeah, it's going to make a lot of bad stuff, but some of it's going to be good. Some of it's going to be better than others. There'll be stuff that's less good, but there'll be a population, a family of viruses that's highly genetically diverse. And because it's highly genetically diverse, it can respond dynamically to a selection pressure. Drug, yeah. Immune system. So the immune system is always chasing this virus. And it's always got a family of viruses that will be resistant to whatever is coming its way. So these mutants, be they drug-resistant mutants or immune-resistant mutants, actually exist before you take the first pill or before you generate an effective immune response. And because of that, it can throw away all the dead ones as long as it makes a few live ones that are diverse enough to make this population continue in an individual. Here's just an example of that. If you go to one particular spot in the HIV reverse transcriptase at position 151, there's normally a glutamine there, CAG. I know you guys memorized it already. One nucleotide change from CAG to CTG gives me a leucine, or CAG to AAG gives me a lysine, or if you make 2 changes, I can go from a CAG to an ATG. Well, okay, all of these will exist to some degree in the population because they all still are reverse transcriptases. These 3 are highly sensitive to all of our antiretrovirals that inhibit reverse transcriptase. This one, however, is not. And this one, and a couple of others with it, is highly resistant. So if this is our population, because it tolerates a low level of something that maybe doesn't grow as well, but it grows good enough, if this is our population, then when you add the nucleoside reverse transcriptase inhibitor, all of these go away, and this one emerges much faster than if you had to wait for it to be selected later. So the integration event, so that's a daunting prospect if we're trying to develop drugs, is that before we ever start, to every single drug, we're going to have mutants available. So we're going to need to do things in combination. We have inhibitors for the integration reaction. This one, probably the best drug to come along in probably the last 15 years or so, that inhibits integration. So far we have inhibitors of attachment, we have inhibitors of fusion, we have inhibitors of reverse transcription, we have inhibitors of integration. After the viral RNA and proteins are made, they're assembled at the cell surface, and after budding, a final event, maturation, has to take place, which is mediated by the viral protease, right? So that viral protease takes things that look like this. As you can see, here's the cell surface, and these are virions budding out of the cell surface. And shortly after that, they mature and look like this. And you can see a pretty striking difference between this, which kind of looks like a donut, and this, which looks like has a solid core. HIV protease digests the precursor proteins that are present here into products, and that turns an immature particle into a mature particle, and it turns a non-infectious particle into an infectious one. So even after the virion is produced from the infected cell, we can grab victory from the jaws of defeat by inhibiting this event with an HIV protease inhibitor. So that's our last set of FDA-approved inhibitors. And if we don't use them in combination, then that pre-existing population, we use them one at a time or two at a time, we use it one at a time or two at a time, we will always lose this battle. But using them three at a time, where it's harder to get all of those mutations lined up in a single virion, we win that battle. We never eradicate the virus because it's established itself in long-lived populations of cells, but we can suppress it for long periods. Here's, I mentioned before, here's how HIV looks in its family tree with 1 and 2. HIV-1 most closely related to the SIV-CPZ. If you look, I'm sorry, I wanted to come back to one last thing and say in human populations we have these HIVs. We also have, and now HTLV, or human T-cell leukemia virus, also one of the co-discoverers here, Bob Gallo. And this virus, how many people have heard of HTLV before? Human T-cell leukemia virus. But you'd heard of HIV before you got here, right? Fair. Okay. How many people do you think are infected with HTLV in the world? This graph, map shows us where all the populations are throughout the world. But for a virus that you didn't really, maybe, let's say 20% of you knew about before you got here, how many people in the world do you think? We're virologists, so we'll do it in log terms, right? 10 people, 100 people, 1,000, 10,000, 100,000, a million, 10 million, a billion. Which one of those would you say? Knowing that HIV is somewhere around 30 million, 38 million. You guys got all of that right. What do you think? I take a guess. What's that? A million. That's a good guess. Considering nobody knows whether it's a million, sounds pretty good. It's closer to 20 million. It's not that far off from HIV. So like, what's up with that? It doesn't have better, it gets worse press. It's got a lousy, manager or something like that. How come more people don't know about HTLV? And the reason is simple, I think. HIV, 38 million people. How many people are dead if you don't treat them? It's a lethal illness with a reproducible decline in death. HTLV is the yin to HIV's yang, where there may be 20 million people infected, only about a few percent, maybe 5, will ever get disease. And there's a number of diseases that it causes. But because it doesn't cause a huge amount of disease, it isn't as big a problem. There's a second virus that's related to it, HTLV, that's HTLV-1. HTLV-2, that's found throughout the world. It's endemic in Amerindian and Pygmy tribes. I don't know how it got there. It doesn't cause any disease at all. So maybe there's only a few million of these throughout the world. So there have been retroviruses in these populations for millions and millions of years. How they got there, probably through zoonotic events. How they stay there is by some détente with the host. So HTLV causes two different kinds of diseases. One is a T-cell leukemia virus. That's dramatic. That's one we study here at NIH. 1% to 2% of all the infected individuals will get it. There's some other diseases that come along with the immunodeficiency that's associated with HTLV-1 infection. But therapy is very, very poor at this stage. HIV-1, back to that. All right, so that little coda is over. HIV, individuals went and looked at the chimp populations throughout Africa and found that there were about 4 main chimp subpopulations, and each one of them has some HIV-like virus. No, actually only 3 or 4 of them. 3 out of the 4 have them. These here have a virus that looks like HIV. The 4th one over here doesn't have anything at all. And it's thought that this population migrated hundreds of thousands of years ago, separated itself, and therefore wasn't around to get infected. As it turns out, and I didn't know this coming from New York, the chimps don't like to cross rivers. And so this made it a relatively isolated population. These were all infected to some degree. And if you put them on a family tree, you can find some that are very closely related to HIV, some that are found more in gorilla populations than in chimp populations. So we can really account for this virus that came into humans by a virus that was in chimpanzees. And how did it do that? It's thought that the poacher was central to this, or somebody who used the chimp as a source of protein. And so, getting these things is not so easy. Butchering them is not so easy. The viremic ones give you ample opportunity to have transmission of blood and body fluids into the human population. They're sold at bushmeat markets, et cetera. And using phylogenetic analyses, we can date the introduction from the late 1800s, early 1900s is likely how the current epidemic came into this population. Now, you stayed here this long, right? Say, well, haven't this been going on for hundreds of thousands of years? And the answer is, yeah. How come we're so lucky? And I don't know the answer to that. Why us? Why then? And I think these are the ways that it got into humans, right? But I think there are other ways that it spread: political, economic, and multiple epidemics. So here's what Africa looked like right before the epidemic probably started. Here's how it looked like when it was up and running, and we didn't even know about it. So there were big upheavals of populations, obviously things being affected by the colonial empires, and the wars, and the malnutrition, and the immunodeficiency that comes with that malnutrition. Another thing, and this may have had more to do with it, or as much as anything else, and that was the population. So here's the population expansion in Africa, God bless you, over the course of the period when HIV emerged and became an epidemic. And you can see down here, once again, we're thinking in log terms here, how there was a vast increase in the population. And the population that has HIV in Africa is not like ours. It's a heterosexual population. So the events that gave us all this population were events for opportunities for this virus to spread. And so I think that of all the things, this increase in population may have had as much to do with it as anything else. Travel in Africa, always difficult, but with development came this Trans-Africa Highway, where people would bring their goods and services across great distances. And because of that, we're able to transmit the virus from place to place, from Central Africa to both coasts, and obviously to West Africa. How to get to the United States is still something of an open question, but there's a clear intermediary that people have found using these phylogenetic techniques, looking at all the viruses in the United States, and comparing them to all the viruses that are like them in Central Africa and those in the Caribbean, specifically Haiti. And the viruses in Haiti are older than anybody else's viruses. So there was a great deal of interaction between Haitian professionals into Central Africa in the 1950s and before, and it's likely from there the virus was transmitted to Haiti and from there to the United States. Once it started to get to the United States, and here's the part that you perhaps remember, the first cases in 81, 82, by 1983, there were 1,000 cases distributed by these yellow dots. And you can see with time, so that's 1,000 cases in 83. By 85, we were up to 10,000 cases, so a log increase. This is an epidemic. And you can see it's still centered on cities. We got the next log from 10,000 to 100,000. The first log took 2 years, the second log 4, and here at 100,000 cases, the next log to 1 million. That's where we are now. Things slowed down after 1989. By 95, we had gotten just to 500,000. And then from there, we're up to a relatively stable virus population. In the United States, about over 50% of the individuals newly infected are men who have sex with men, about 30% heterosexual, and then injection drug use, about 12%, and then the combination here. And I don't know how these numbers are going to change with the opioid crisis. This is something that really has to be investigated. But one thing that seems to be true is that it's becoming much more of a heterosexual population that are getting this virus. Women comprise now greater than 25% of all AIDS cases. And in the District of Columbia, which has the highest prevalence of HIV in the country, women represent the fastest growing population with HIV. The epidemic is aging. About 11% of all cases now are over the age of 50. And about now, 50% of cases in the United States of HIV infection itself are over 50 years. Well, how to treat this, right? This was a poster that was on a billboard that was on the side of buses driving around Washington, D.C. about the time when antiretroviral therapy was beginning to be introduced. If you got the AIDS virus now, you and your license could expire at the same time. The idea was get tested, know, and then that was the only way that we could have to block transmission. Over that time, there were some new posters, right? And the one that's most important, I think, now is the catchphrase 90, 90, 90. How many people have heard that? 90, 99, it's big, 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 right? 90% of the epidemic should know their status. 90% of them should be engaged in care. 90% of that total should be suppressed on antiretroviral therapy. So the idea is treat everybody. As long as we can get 90% of everybody who's infected to know their diagnosis, and 90% of them engaged in care, and 90% of that 90% suppressed on therapy, we're going to get to zero. So 90, 90, 90, treatment for all, getting to zero, was started with AZT in 1987. So this timeline tells you when the drugs were first FDA approved. They're color-coded by whether they're inhibiting reverse transcriptase or protease or co-receptor, or integrase, or inhibiting fusion. And here in that first couple of years, it's one drug, it's another drug, it's another drug. None of these could suppress the virus because we couldn't use them in combinations of 3 or more until about 1996, when multiple drugs, multiple targets suppressed HIV below the limit of detection. So this takes us to 2001, right? After that, we have the development of integrase inhibitors and now better drugs, ones that are better tolerated with longer half-lives. So we don't have to take a handful three times a day, one pill once a day. World AIDS Day is coming up. You'll see it in the news a great deal. There'll be a report from the District of Columbia Department of Health saying how much better are we since last year as to how many newly infected cases there are. Last year, I think it was 376. This year, it's going to have leveled off. It's in the 370s. I don't think it will have gone down. But look for that, and there'll be a remembrance worldwide. A lot of information about how the world is doing. 37 million, half on therapy. All right, that's not 90, 90, 90, but there are countries that are coming close. If you remember that Gardner state of care, how are we doing? Well, the first 90 were not so bad. The second 90 were not so good. And the third 90, there's a long way to go getting people engaged in care and getting people suppressed on therapy are where our biggest issues are. So the need is great. The prevalence is increasing. R90, meh, we're getting close. Here's how many are infected. Here's the first 90. Here's the second 90. Not close. And here's the third 90. 300,000 out of this total. So we're about 75, 50, 30 is my guess. Key advances since that now are therapy for those people who don't have infection but have a high risk of being infected. Let's give them the antiretrovirals and prevent transmission. We need to continuously give therapy to avoid AIDS and other complications. The earlier the therapy is initiated, the greater the preservation of your therapy. Vaccines and cures are still things that neither one of those that we have. One person has been cured by HIV, Timothy Brown, in the context of a bone marrow transplant. That's not a feasible alternative for everybody else. So lessons, viruses are bad and they should be avoided, except maybe when they save the planet, right? And maybe if it saved you from the last virus. So our TRIM5 alpha, I think we should thank it for all of its nice service in the last epidemic and be grateful that we don't have to wait for us to evolve to be resistant to this epidemic. Epidemics are not single events. There were multiple introductions of these viruses into human populations. Those epidemics evolved now. So whereas at one point we were increasing greatly, now we're at the state we're increasing slowly. And in understanding of replication clearly led to new therapy that saves lives. Antivirals are useful, instituted as early as possible. Adherence is essential. Stopping therapy means resurgence of virus replication. So I'll stop there, and sorry to go over by a couple of minutes, but if there are questions, I don't know if there's time.
With cancer, the problem is the patients often become resistant to drugs.
Right.
With HIV.
They were way ahead before we even started. So they don't get resistance if we use enough drugs to suppress them. So there are very, very few examples of effect, and I don't know of any, where the therapy was as effective as it should be and the virus became resistant. There's a huge debate as to whether there's any ongoing replication that goes on during this suppressive therapy. And the work that we've done here suggests that there isn't. Other people have suggested there might be in sanctuary sites where the drugs don't penetrate so well. But that isn't something that we found. When the drugs don't penetrate well, resistance does emerge.
Why are there people who know, why don't people accept the treatment? So why do the chart you have created?
This one.
Yeah,
So like, what's up with this, right?
Where how can you increase the
Right. On the one hand, these are our failures. These are our challenges. These are our opportunities. So people that get diagnosed, well, where do they get diagnosed? Everybody goes to the doctor, can get an HIV test, should get one every year. And people who have higher risk, some people say they should be tested more frequently. Other testing goes on in the setting of an emergency room. Somebody comes in sick, they get an HIV test, they're supposed to get them everywhere, and they don't. But the stigma associated with being diagnosed keeps them from coming back. So even though we can confirm a diagnosis in an emergency room, well, what are you going to do then? You'll say, well, listen, should we start antiretroviral therapy on the spot? There are studies that are studying that. Should we refer them to engage them in care? Because somebody's going to have to refill this prescription. Well, that happens and people don't come back. They're scared, right? The follow-up isn't all that good. People don't go out and get them, right? For tuberculosis, we've got a great therapy for tuberculosis. And we treat people with directly observed therapy to make sure that it doesn't spread. That might be a good idea, but there are many, many more people who would have to be engaged. And so it requires that people come in and engage in care. And the dirty little secret is that, and I've seen this a few times, right? People start therapy, and you know, we're such nerds at NIH. All right, maybe Terry's not a nerd, but okay, some of us are nerds, right? And people come for therapy, and you show them their data. And what do you show them? A very high viral RNA level. It's 100,000. Very low CD4 count. It's 20. You start this therapy, and a couple of weeks later, the viral RNA level is really, really low. And the CD4 count's coming up. And you can draw a graph, and it looks terrific. And they're, you know, sometimes they're just being patient with us and not telling us that the pills are terrible. And I'm just barely making it. And we're so excited about telling them how great they're doing that we're not listening to them. And so sometimes they'll stop. They don't tell us because they don't want to make us feel bad. Or they do tell us, you know, months later, nothing's different. The CD4 counts come up. It's not getting any better. Viral RNA level is down. Not getting any worse. We're in a stalemate. What do we do now? They have to take the pills every day. And that becomes a difficulty. But that's retained, and that's when they're retained in care. On therapy and staying on therapy, we're still making, here's still a challenge to keep people on therapy. Here's a challenge once they've been linked in care to get them retained and on therapy. Because the worst of it is starting therapy from the resistance standpoint and saying, yeah, I'm all gung-ho about it. They are. And you don't blame them. And then down the road, they stop coming because they feel good. In fact, when they stop that therapy, they feel a little bit better because the virus hasn't come back. And so that engagement and linkage and keeping people in care and doing it right is where we're falling down on the job. So there's a challenge and an opportunity for that. But there are a number of different reasons, stigma being one, fatigue being another, and I don't know, people lose their care, travel, things like that are other reasons. Yeah, cost is an important issue. And so the ACA covers it, but not every plan covers every pill. And so some of them, cheaper plan, will pay for a twice-a-day drug like raltegravir, an inhibitor of integrase, and maybe not for the dolutegravir. Sounds the same, take it once a day. It's probably a better drug for that reason. So yeah, no, that's absolutely true. Is it $10,000 a month the way it used to be? Not anymore, because insurance will cover it. And for people who don't have anything, there are ADAP, or there are AIDS programs that will support their therapy. So yeah, there are donut holes that prevent people from getting some complex therapies, and that's a bear. But then, you know, there are other ways to try and advance that. That one's probably getting better. Whereas over here, we still got a challenge.
Okay, that'll do it, Frank.
Yeah, I'm sorry to keep you all